Yabing Chen is a new recruit to OHSU, arriving towards the end of last year. She's currently the professor and vice chair for research in the Department of Pathology and Laboratory Medicine. Prior to that, she was at the University of Alabama, Birmingham, where she was also a professor and vice chair for faculty development and education. And she also had a position at the Birmingham VA as well. You have a position here at the Portland VA?
Yeah.
Yeah. So she moved with a whole bunch of her lab. She has several R01s that are currently running and several VA merit awards. And so a very productive lab, and this would have been fun. And so her interests are in vascular stiffness, which of course is of interest to a bunch of people in our institute. And one of the goals for this is so that we can all learn about her work, meet her, because she's always looking for opportunities to collaborate, mentor, etc., and expand horizons. So without further ado, go ahead.
Thank you. Thank you so much for the invitation. Is the voice okay? Okay. Like Matt said, this is really a great opportunity for me to learn this institution and present my work looking for collaborations. And over the past probably 15 years, we've been working on vascular stiffness classification. And actually, after so many years, we start to think about it this is a problem of pan-vascular, not isolated, because our study have been focused on cardiovascular at the beginning to start with, but now we're looking everywhere in terms of vasculature and how the pathology in one place may affect by other vascular changes. And of course, for this audience, I'm sure you know aging is the biggest factor for all the cardiovascular disease. And let me see if I can get this laser working. This is a cardiovascular incident and cardiovascular death. Yeah, good, it works. But probably you're not fully aware of this situation. Actually, this physician said 300 years ago, a man is as old as his arteries, and I'm sure we would appreciate more. This is a situation if we have healthy vasculature, blood vessel, and probably that reflects the life quality and the life experiences. So during the aging process, there are many changes. Particularly, I want to use this cartoon to highlight the changes in the medial area where the intramural expansion could happen. And that's associated with lots of changes within the cells, especially the endothelial cells and the smooth muscle cell. That's where our study most focus on is the smooth muscle cell. And that leads to intramural thickening. So that start to expand the thickness and also lost the contractility. So that's why it's more stiffer. What you can see, a few highlights of the disease associated with is, you probably heard, is hypertension, atherosclerosis, calcification, and stroke. And our study mostly, in particular, related to mechanistic study is focused on calcification, as this is a very devastating pathology that happens in a lot of disease conditions. I use in atherosclerosis, diabetes, and stage disease is what those renal failure patient needs dialysis. This is a very devastating situation. If you can see here, this aortic valve calcification, if you have calcified nodule in the valve, they make it very hard to close and open. So what often associated with a regurgitation, the blood vessel, blood is kind of reflects back and make it hard. Your heart have to work harder to get the circulation going. And here is another example what's happening in the coronary artery. So that's the artery around your heart. So provide nutrients and blood to the heart, the cardiomyocytes. If that's occlusion and you have the impaired myocardial infarct, that's heart attack, what we often refer to. And calcification actually has been recently demonstrated to be an independent risk factor, predict a cardiovascular outcome. And you might think, okay, this is an old people situation and I might not have it, but here is the study showing in age 30 to 39, there's already 21% of women have, men has calcium deposition or calcification in the artery, and the woman is more protective, and the next age group, it kind of double up. So that's one of the situations we should be aware of. That's, I often say, if you haven't got your calcium score tested, better ask your cardiologist or your physician to see if that may work out for you. Another example here is showing how devastating with increased age, you see increased calcification. This is a peripheral vasculature in the, not in the coronary artery. So you see a lot of dark black staining, that's calcified area, but compared to healthy normal vasculature. But the problem is, so far we have no treatment for calcification. Often you heard is treat the underlying disease like lipid lowering and calcium, glucose controlling, and that could be helpful for the calcification development in related to a different underlying disease, but no direct drugs can treat it. And the other part is when we do not have therapy, what about intervention? That's what so far been successful use is you heard about the PCO angioplasty. When you have occlusion, they use a balloon to open it up, so make sure that the blood can flow. And this is what happens. It's caused injury to the vessel as well. And you see this layer is smooth muscle cell proliferation, and migration eventually cause thickening of the inward. So go inward into the lumen, and we call that restenosis. And the other part you probably have been aware of a lot is stents. When somebody's grandparents have some blockage issue, they put in a stent. It's like a spring, put in and open up. And what happened, I will show you an example. The stents were not cure or reduce calcification as well. And recent advance in technology also have all this device that can help to get rid of those calcified area, because they are hard, they're thicker. So you probably need to look at all those the loader bladders, and this is a sonication and a laser. Try to break it down to hopefully that can reduce they can kind of fresh out or shave out. They can go through the circulation system, eventually get rid of them. But that's a lot of problem with that as well. So far, that's no direct either direct treatment or intervention can help with calcification. Here is an example I mentioned earlier about calcified artery. This is coronary artery from patient with the stent put in. So here are the remnants of the stents when you cut a cross section of that. So you see the metal is still there, and this calcified area is still there. So this patient died 2 years after put in the stent, and you probably can appreciate this in-stent restenosis is a thickening going inside. And another surgery we often heard is the bypass. When the tissue is damaged, the stent didn't work, you have to cut it off, replace it with a new one. And oftentimes we put in a vein, so from the leg or from somewhere. So that when the vein normally they're not calcified. So in any situation, they may not calcify. However, when you put the vein into where the arteries is, you start to see this calcification lesions. So this is a failed bypass autograft. So that will not work. And that's why we have to go back to understand what's going on, why this is happening. And here is how we have been trying to understand about what happened to the pathogenesis of calcification. It's mainly the atherosclerosis-related calcification showing here. Is you can see it's more asymmetric associated with atheroma. And here purple stain is where the calcified area. But in the media calcification, which is often associated with diabetes and the renal failure patient, they're continuous across the vasculature. And the common feature of this too is smooth muscle cells. They can become calcified, they can become a bone-like cell. But also we would like to know is smooth muscle cells are highly heterogeneous. Here it's showing basically is different origin of the smooth muscle come from during the development. And those cells also very highly plastic. So they can transform from a smooth muscle cell, the contractile, that's how your blood vessel is contract, because the smooth muscle have the function. That's their main job. However, in disease condition, they become thicker, they lost the contractility. And they can also become a bone-like cells, because that's a de-differentiation and the trans-differentiation of the smooth muscle cell to other phenotype, cell type, like a bone. They can engraft lipids as well, like a macrophage-like cells. So over the years, we started to understand that this process is actually mimic the bone formation. They use a similar pathway like a bone cell form that derive from mesenchymal progenitor cells, acquire all those markers, and they become osteoblast, that's a bone cell, or chondrocytes, that's the cells form your cartilage. And the difference between these two is this is mineralized, but the cartilage, they are not mineralized. And smooth muscle in normal situation, their extracellular matrix is not mineralized. But during the aging and disease condition, they start to acquire those features, the markers for bone cells. They have the mineralization in the extracellular matrix. So that's why they become very stiffer, function like a bone cell. And over the years, our study have been focused on this transcription factor called RUNX2. As shown in the previous slides, is critical for the bone formation. However, during the calcification process, we and others have been shown using different models, even in humans, in animal models. Here showing the cell culture, RUNX2 is go up associated with calcification of smooth muscle cells. We have diabetic animal models showing RUNX2 is elevated. And in atherosclerosis model, this is a calcified area showing in red, and the green staining demonstrated RUNX2 upregulation. And similarly, in human disease condition related to atherosclerosis, we see RUNX2 upregulation in this calcified area, and also in the coronary artery from diabetic patients that go up. So a simple question my student asked when they this is my first student, and when we start the lab, and we ask is really a determining factor for RUNX2 to drive the smooth muscle cell become a bone-like cells. So the experiment he did is increase expression of RUNX2 in smooth muscle cell. This is in normal, isolated mouse smooth muscle, they express very minimal RUNX2. However, when he increased the expression, enforce express RUNX2 in those cells, they start to become calcified. So that indicated RUNX2 is the driven factor, cause the smooth muscle cell become a bone-like cells. So with that information, and then we said, if we knock out, So if we take the RUNX2 away from smooth muscle cell, would we able to prevent calcification in the animals? So we did a tissue-specific knockout for RUNX2 in smooth muscle cells, and then we isolated cells and confirm that delete of RUNX2 prevent the oxidative stress-induced calcification in cell culture, and also in atherosclerosis model, we use high-fat diet to feed the animal to make them more prone to develop atherosclerosis and the calcification. And with the smooth muscle specific RUNX2 knockout, we were able to block or at least markedly inhibit the formation. But what made us more confident about this observation is two other labs, they generated independent smooth muscle specific RUNX2 knockout, and they also had a similar observation is delete RUNX2 in smooth muscles inhibit calcification in this vitamin D induced rhinopharyngoma model, or in atherosclerosis. They use a different model for the atherosclerosis. And this is a more recent study similarly confirmed in the disease condition RUNX2 go up, but is associated with calcification. But when you have RUNX2 deletion, that calcification was prevent. So this all study from different group using independent animal model confirm if we block RUNX2 in smooth muscle cell, we were able to inhibit calcification. So during the study, my student also had an additional observation. So what you can see here is atherosclerosis formation. The red is the oil red or Sudan staining, indicating they were atherosclerosis lesions. You may appreciate either when high-fat diet feeding, you get more atherosclerosis. But in the RUNX2 knockout animals, either in chow diet, the normal control, or the high-fat diet, the development of atherosclerosis was markedly inhibited. So oftentimes we were thinking the process is develop atherosclerosis, all the information, all the other thing leads to calcification. You often see in the advanced stage of atherosclerosis. But this is kind of indicating they may have additional function prior to the calcification process. That's the part we're actively investigating. But we also did other models, like what you see in the restenosis model, the thickening of the, so we cause injury in the artery. And then we saw the thickening of the intramural area, but with RUNX2 knockout, that was blocked. Another disease model we use is the aneurysm model, which is a blow up of the abdominal or thoracic arteries. And this part we use angiotensin II to induce the formation. And that is, angio-2 is a also induce hypertension as well. However, in our knockout animal, even we infuse angio-2, the formation of aneurysm was blocked as well. In our recent study, this is, sure. The angio-2, yes. So I think originally the angio-2 without with the B6 background, they developed very slow. And with the ApoE, that's probably what's in the field. They found out that model is more prone to, they can develop disease quicker. Yeah, but definitely one of the mechanisms we learned is the information. Yeah, yeah. But even with the information in the condition, in the control, IL-6 was go up, but with the RUNX2 knockout, we found IL-6 was downregulated. The angio-2 induced IL-6. That means smooth muscle cell RUNX2 may have other function in addition to driving the bone formation. And lipid. It, for this short period of time, they did in 28 days. They do not. Yeah, they do not. At this age, so we normally use in 8 weeks old, 8 to 10 maximum. And then they are normal child eye. And they only, yeah, put NG2 into them. Yeah, thank you. Any other question? So, yeah, go ahead. No problem. They, the normal size, yes. I will show the artery and later to try to understand why this is going. Yes, they develop normal, and their vasculature anatomy and the smooth muscle expression is similar compared to. So another recent observation we had is collaborate with someone who studies cerebral vasculature. And what we found is in the RUNX2 knockout also have improved the cerebral vascular perfusion here. Normally, that would be associated with the cognitive function. So we put those animals in water maze or open theater to see if they have changed their behaviors. And apparently, the knockout took less time to find the platform, and they seem to move faster. So that means they may associate with their memory or other cognitive function. So this is another area we're active investigating. But since we got here, yes. So those were with HIFA died for 6 months, so they are 8 months old. Yeah. And one of the advantage here when I moved is we got this brain tissue bank, and I took advantage of them and look at the RUNX2 expression in the brain. And to my surprise, they are not only expressed in the artery, the vessels, blood vessels in the brain, but also seems to express in other cell type as well. So we look into one of the literature. This is I think a single cell mapping of RUNX2 in the brain vasculature, and they found this not only in smooth muscle parasites that's related, but also in microglia and other astrocytes, oligodendrocytes. I think that's the area we look most here. So that means RUNX2 may function outside the vasculature and then contribute to all the disease conditions we have seen so far. So that's why we think this is an important area we're trying to put some effort to kind of extending this observation. So with all these animal models, apparently they all accelerate or associate with aging process. So we were wondering how vascular RUNX2 may contribute to the aging process. And simple experiment we did is compared the arteries 2 months old versus 18 months old. So that's a mouse study. And we definitely identified increased RUNX2 protein here in the mouse artery that was associated with thickening of the arteries. And we did pulse wave velocity to look at the stiffness, the function, and we found increase in the 18 months old. And gradually looking the growth of this RUNX2 expression in the artery from 2 to 16 months, we found all increase gradually in the mouse arteries. So the question we ask is, okay, in the blood vessel, what's the function? What really RUNX2 contribute and how that regulate the signaling pathway, in addition what we know about the promoter, the calcification. And we took advantage of the single cell technology and analyzed the artery from this is mostly smooth muscle cell lineage. And we found in this knockout animal, so those are the smooth muscle cell cluster from the knockout animal, we found much less of the modulated smooth muscle. And I mentioned the smooth muscle cells, they can change their phenotype. So they came from the contractile smooth muscle cell and de-differentiate from and transdifferentiate to other cell type. So this process is called a smooth muscle phenotypic modulation. And all these modulated smooth muscle cell, they were originally derived from the contractile smooth muscle cell. So when we have a RANKS2 knockdown, the smooth muscle cell are much less likely to become a modulated smooth muscle cell. And they actually, mm-hmm.
And that kind of made me wonder, is there some of the pulsating velocity changes or sensitivity changes because some muscles that may still be contractile are not able to like conduct to each other because they're further apart?
That's a possibility. Yeah, because you have thicker arteries. Then when we do pulse wave, is looking the we look to catheter in two areas and then look in the distance they traveled and the velocity. Yes, the thickness may be able to affect it, but we do not know how. We cannot parse out the contribution of the thickness to the pulse wave velocity we see here. But definitely we can see RANKS2 upregulation is across the entire layer. Yeah. So that's a great question. And related to that question is, where are all this smooth muscle come from? Are they residential smooth muscle proliferation, expression, and migration? Or they're from cells from the adventitious. Those progenitors, they also can travel into the intimal end. So we probably need to do more work to trace where they come from. Yeah. Okay, back to the single cell data. What we found is RANKS2 knockout in smooth muscle cell inhibit the transdifferentiation, but maintain the smooth muscle cell in the contractivity. So that's how the population, if you see here, knockout have 80% smooth muscle lineage, the original contractile, and the control, they had 50, means 50 already modulated, transdifferentiated. And when we look more closer about the signal pathway, they are more related to extracellular matrix organization and information. So that's two pathways we're focused on. How RANKS2. For those who be made interested in this transdifferentiation, I can go further, dig into the data, how different modulation of the smooth muscle cell layer. But back to your question, what happened to RANKS2 knockout? With the affected growth, with the affected development, they seem to comparable to their wild type or the flux control mice. And also the cross-section seems to be similar in terms of the structure and the smooth muscle marker gene expression. So the question is, what happens during the growth or during the disease formation? I have mentioned the age is a dominant factor. So during the aging process, that's a chronic effect. We have all the stress over life, probably that's why. And there were some genetic factors affected, and I did not have a chance to mention some of those. You probably, ApoE is one of them, because in the ApoE knockout or LDL receptor knockout, they have a high incidence of have tissue get calcified. But we work in environment health, so the modified factor probably is more related to what we know for the general population. Is there many of them is hypoglycemia. That's probably why in diabetes, calcification is a devastating pathology. And hyperlipidemia-related atherosclerosis, chronic kidney failure, and the stress, also the lifestyle. I even talked, Matt, we recently have data showing the BMO, the circadian regulation knockout that can have an impact. I'll probably show one slide on that because that's under revision for circulation. So hopefully, fingers crossed for that study. But what I want to use this diabetes condition as an example to show how we use this to learn the mechanism and the regulation for RUNX2 upregulation and calcification. And what we have focused on is on this glucose metabolism here, showing go all the way down, looking, this is called hexasesamine biosynthesis pathway. And what this pathway happened to is in the normal condition, probably 5% of the glucose go down this way. However, in diabetic condition, they are elevated up to 20%. Depends on the situation condition. And that leads to the generation of this UDP glycanic. So that is a sugar donor that can be added onto proteins by the glycosylation modulation called O-glycanic molecule, because it adds to the O-link serum threonine of the amino acids on the proteins. And our focus on this one is, like I mentioned, they go up. That process going up during the diabetic condition, that can be associated or maybe cause the pathology in diabetic vascular path. So the first experience my student did was, okay, we kind of predict, we have diabetic model and the glucose metabolism through O-glycogenation may go up, but is that happen in the vasculature? So he isolated the arteries from the STZ induced. This is a mimic type 1 diabetes causing hypoglycemia. And we found increased O-glycanic in the artery from the STZ treated mice. That's associated with RUNX2 upregulation, calcification, and the stiffness of the vasculature. And when we look more closer, in the arteries, the O-glycogenation is definitely showing consistent with nuclear localization, and that's associated with decreased smooth muscle markers and increased RUNX2 expression. So what, yeah. So far, our data indicating is in the nucleus. Yeah, yeah. There were some forms of mitochondrial OGT, but that is not in our study. And if you look at the staining, what we found is mostly in the nucleus. Yeah. So again, we want to verify that the one I showed, increased calcification in the RUNX2, whether that's associated with increased O-glycogenation in that same patient sample. So what made our life a lot easier is this process is controlled by two enzymes. One is called the OGT, it's a transferase, adding the sugar on, and one is called the OGA, that's the enzyme remove the sugar. So we first mimic the diabetic condition by inhibiting the removing. We predicted that will be increased O-glycogenation in the cells. So we use both the pharmacological inhibitor showing here and the shRNA for OG. That's a review ask. We have to confirm we cannot just use the inhibitor. So we did both, and definitely you see increased O-glycogenation when we block OGT function, and that's increased calcification showing here, and the RUNX2 expression. So the other side of the story is OGT. So if we block the OGT, that's remove the transfers. So then we'll have less oligocyanation, as you can see here. And that was associated with inhibition of calcification in the cell culture. So of course, the most important is how would that happen in vivo? And long story short, this is a really long breeding for this animal because OGT is expressed in X chromosome, and the smooth muscle specific induced Cre is Y-linked. So it took us a long time because we cannot straightly delete OGT in smooth muscle cell. If we delete that, the mouse will not survive. So we have to let them pass the developmental part and then induce only when we challenge them with the STZ or other condition to induce diabetes. So, but the good story is, ending is we got those mice, we were able to use the male mice and they are control little mice to do experiments. And what we found is definitely when we have OGT inhibition here, and then we got less oligocyanation and the inhibition of RUNX2, that was associated with decreased calcification, improved possible velocity. And here another evidence showing RUNX2 expression mRNA is downregulated in the artery. So this is another paper we're currently under review. So we'll see how that one will go. Then go back to the signal pathway. So what happened? How this is overall signal pathway affected the RUNX2 upregulation. And our early study have shown OGT as a critical, AKT signal pathway is critical. And in this study, we found that AKT itself can be oligocyanated, and that can affect the AKT phosphorylation. And for those who study AKT signal pathways, AKT phosphorylation is critical for its function. And if we block the AKT phosphorylation, that was linked to decreased calcification in the RUNX2 function, RUNX2 transactivity. And I mentioned the AKT is critical when we had a study early for RUNX2 upregulation. And if you can see here is when we block AKT phosphorylation with this inhibitor, we were able to block calcification and the RUNX2 upregulation. So downstream AKT, we identified this FOXO transcription cofactor is very critical. And when we have AKT phosphorylation increase, that's sufficient to induce calcification. And what happened to that is AKT can phosphorate FOXO, leads to FOXO nuclear exclusion. So from nucleus to the cytosol. So when they were there, they lost their transcription activity because they have to be in the nucleus to active. So that nuclear exclusion was associated with RUNX2 upregulation. And we further teased out that it was through the protein ubiquitination of RUNX2. Ubiquitination normally is associated with degradation. So we have decreased ubiquitination of RUNX2. So the RUNX2 will not be degraded. So it's accumulated. So that's how is RUNX2 is accumulated in the cells and it leads to calcification. So we get excited if we have AKT. Yeah.
Does that mean there's just pan loss of ubiquitination? Are there also other proteins within that?
No, this is specific. Yeah, specific is because FOXO regulates E3 ligase. That is critical for RUNX2 ubiquitination. Yeah, yeah. So, and we know which one it is. Then we're trying to see how specific would that, if we knock out that ubiquitination, would that be sufficient to affect calcification? Right. And so, like everybody, now we got an inhibitor, and we know that's an inhibitor available for AKT activation for cancer treatment. So we say, can we test that in our system? But when we put them in our system, to our surprise, that increased calcification. And then we later learned is, yeah, it inhibits AKT phosphorylation, but it markedly increased P38, which is another signaling pathway. And P38, we found, is critical for RUNX2 phosphorylation. So when we knock out P38, the RUNX2 phosphorylation or RUNX2 expression was inhibited, and the RUNX2 activity was blocked. So that leads to inhibition of the calcification. So P38 is critical to phosphorate RUNX2, and that phosphorylation is important for RUNX2 transcription activity. So majority of RUNX2 function is fine-tuned or regulated by its post-translational modification, the PTM. And the other thing is unique about RUNX2 is a transcription factor. It can bind to its own promoter to kind of induce expressions. Once you go up, then you kind of have a vicious circle to lead further RUNX2 upregulation. So again, this didn't work. And we end up finding additional regulation for RUNX2 upregulation. And a recent study, but confirmed that O-glycogenation can be activated not only glucose, but also impair the intracellular calcium signaling. That can lead to O-glycogenation change, basically upregulation. Eventually, that contribute to RUNX2 upregulation and smooth muscle calcium. So this is a sensor. So O-glycogenation is an intracellular sensor to the environment change. Glucose, calcium signaling regulator, and lipids sometimes will affect that as well. So that's why I think this is a unique condition that we can target possibly, or at least we can use as a biomarker as a signal for that will lead to vascular change. So, and then one last thing I want to mention here is RUNX2 itself. That's a recent study. We found that RUNX2 itself can be O-glycan modified, and that's contribute to its function. When we have knockout this OGT, then we inhibit RUNX2 transcription activity. So not only the expression, but also its activity. Okay, so back to the slides I mentioned about circadian, is what happened to diabetic patient. This is one of recent study in human, looking the glycemia change over the circadian clock. And you probably can appreciate that the drastic fluctuation in this diabetic patient compared with this control. And what we learned in our study is in our STZ treated animal or in high-fat diet treated, the high-fat diet induced obesity model, we found O-glycogenation is going up, and that BMOR expression is going up as well. BMOR is one of the key regulators for the molecular clock. So for those studies, that's a transcription factor, and it binds to clock and regulates the PERT and other circadian regulator gene, eventually feedback to contribute how your 24 hours will be different. But what we found in this chronic condition is in diabetic condition, they can lead to BMOR sustained go up. And then the question is whether that's mimicked in the cell culture as well. So we looked the glucose level and treat the smooth muscle cell. We found the BMOR 1 definitely is going up, similar to the observation in vivo, but not the other circadian regulator like BMOR 2 or clock. So we went on to generate smooth muscle specific knockout animal. We took out the cell to do a sequence analyze, and you can see a lot of this signaling is associated with a smooth muscle cell proliferation, differentiation, and phenotypic transition. And when we have knocked out BMO, we found the RUNX2 upregulation was inhibited or at least partially attenuated. And when we took out the cell and did the calcification, what we found is BMO deletion can greatly attenuate the calcification, especially in the media area, because we do smooth muscle cell calcification. So over the years, and one of my goals is looking for inhibitors for calcification. And oftentimes we found those activators. But I do want to end with a good news is we did find this dietary potassium, which is helpful, at least in these animal models. This is atherosclerosis-related calcification, and the lower increased calcification, the higher potassium supplements that inhibit calcification. That's associated with RUNX2 inhibition, and then we found that signal pathway is through autophagy regulation. So with that, I hope I have convinced you RUNX2 is the factor, very important for smooth muscle cell once you have the upregulation by all these factors, and that can cause damage to smooth muscle cell, including mineralization, matrix deposition. And I didn't show you data and that affects synapses as well. That's associated with all this disease condition associated with aging and others. Okay, I think with that, that's currently my team come across the country with me. So we currently have 4, and I actually had 2 new postdocs join us this month. So it's like 6 people, 7 people now, and we look forward for further collaborations. Yeah, thank you. Good.
So that sex specifically, is that due to changes in this process, or is the RUNX2, for example, higher in men?
Not really, and I think that's other factor coexist with RUNX2 upregulation, and that's our preliminary observation. Is smooth muscle cell for male mice and the female mice, they have different potential for differentiation into bone. And they do express collagens, and all those seems to be comparable. But the last part, the mineralized part, is different. So probably that's why in some case, calcification is more often seen in men, but fibrosis is often seen in women. So that's that area we're trying to figure out, but it's difficult because in cell culture, we can control the different steps, like a proliferation phase, mineralization, matrix maturation. And the last step probably is the mineralization and all those. At that stage, RUNX2 actually is going down. So we're trying to figure out why they are different at the last step. Yeah. Yes?
First of all, elegant, really nice story.
Thank you.
In your various mouse models, have you looked at either wound healing or neovascularization, or say, if you were to transplant a tumor, look at those considered sort of pathogenic vascularization in tumors, so that would your RUNX2 knockout then have a very different response in terms of, say, anything that would be associated with neovascularization?
So we did not do that. Especially you mentioned wound healing. Actually, we tried to collaborate with one of my previous colleagues, and she did some experiment that she look in the mice. You kind of punch something in the skin and look at the growth. She did some experiment, and the RUNX2 knockout was effective in prevent. But for the tumor part, we haven't done anything in terms of, because a lot of study in tumor, especially the solid tumor, they found RUNX2 expression is going up in the tumor compared to the environment tissue. But I would suspect, particularly related to what we learned about RUNX2, can regulate some inflammatory factors. They can crosstalk to that's a good thought. If you're interested in looking at that part, we can definitely give you our animals.
But I was also wondering that if you just do something super simple like a sponge pump, where you just implant a small piece of sponge in the ear and then monitor the relative improvement of vasculature to that sponge, then it's much less subjective than, say, wound healing from incision or things like
Okay.
that. It simply is something that is sitting there calling to be vascularized.
And you can use adult mouse to do that?
Yeah.
Oh, that's wonderful. So if you have protocol approved, I'll be happy. I'll be happy to.
The approved protocol lies with other people, past life. But the other question I have is that in terms of do these animals have, say, if you run a glucose tolerance test on wild type versus, what is the response or
Similar.
changes to GTP?
Similar. When we challenged them with STZ and we did glucose test, ITT and GTT, they are similar. The glucose level is go up as well because we're doing tissue specific, not much contribution of those from the vasculature.
I was fascinated by the capacity. Is that bad?
We put them in food, yeah.
What happens with that with calcium supplements, say somebody who goes to
Don't do that. And also, although they will oftentimes say you do the vitamin D combined version, but still, do not, please. Yeah, osteoporosis often, I don't think it's a calcium deficit for patients with osteoporosis, and they often have increased calcification in the vasculature. So it's an imbalance of calcium. So probably you need to treat the osteoporosis, but that's more, I would suggest treatment on the osteoclast instead of make more osteoblast formation. In one of the study, I'm not sure if you're aware of those, they treat dinoxidil, which is osteoclast inhibitor, the antibody, for post-menopause women, average 84 years old. They already have a severe osteoporosis. They treat that to block osteoclast formation. Those are the bone resorbing cells. That was effective to prevent the progression of osteoporosis. However, their calcification was not, in general, the overall calcification was not affected. But if you separate the data with somebody who started with high calcification versus low calcification, the drug was effective for those people who have low calcification to start with. So that means that's a treatment window. What we found is smooth muscle cell, they crosstalk to the monocytes, they're the progenitor cell for the osteoclast. So those are resorbing. And probably it's similar in the vasculature. They respond to the injury, like you injure a bone. What happened is osteoclast will come, clean up the surface. They secrete some factor. Then the progenitor cell will come to repair. In similar situation, I think happens to the vascular as well. So they will have osteoclast come trying to, oh, this is not normal here. I have to clean it up. But what happened is similar to what happened to repair of the bone. The progenitor cells see the signal, okay, so you are clean those up, I need to repair. So that worsens the situation. Probably that's a treatment window when you identify the calcification soon enough before it gets worse.
Yeah, so I'm really not interest, like usually the open macronutrients
Yeah.
with runs too. Are you finding residue of open macronutrients on runs too? And like, I guess these interesting we thought like intracellularly how this is working. Like a lot of my understanding is like neural but functional changes will help on things to it's very sticky ECM.
Yeah.
Is there like do you use the stiffness coming from like changing the intracellular composition for thicker details? Like I don't know, like
Can you see this one?
yeah. It is also like yeah.
Oh, how can I okay. Anyway, so if you see this, yes, we found the residue on RANKS2, and we have did specific site-direct mutagenesis and identify this 412 threonine was critical when we were replaced with alanine and block O-glycogenation or inhibit overall O-glycogenation of RANKS2, and that was associated with inhibition of calcification. And we also generated an animal model with this site direct, like knock in this mutant. Yeah. Thank you.
In patients that have a surgical procedure to put the vein in place of an artery, do you then follow those individuals over a period of time? If there is significant calcification continuing to occur in the artery, and you say, look at the vein, is it also susceptible, or is it in fact resistant to the calcification?
For the grafted bands.
Into the context of market.
That's a good question. I never thought about it. So what's your thoughts?
I just have a question. But it is, let's just say that it has to do with relative, everything keeps relating back to reactive oxygen species,
Right.
information. So if you end up having an environment in which the oxygen is being depleted in the vein, therefore you're not having that kind of stressor all the time. But now if you put the same tissue in the environment,
Yeah.
you are continuously at a relative elevated dissolved
So that makes them more susceptible to.
would you potentially be under the same stressor of them being susceptible to moving toward the calcification?
I'm more leaning to this, this hypothesis, and also the shear stress is different. Like you mentioned, in the vein, they are very low pressure, and when you put it in the artery, the stress is much higher. Yes.
So
Thank you.
It seems that enough people have this that if one did autopsy, you would potentially be able to go in and retrospectively look to see whether the vein remains, let's just say somebody lives 20 or 30 or 40 years post that treatment, then if the environment is driving that increased calcification, then you would end up seeing the vein also experience an increased calcification, but if it was essentially resistant, then one might think that it had not arising from, say, something simple as
Okay, I will go to Dr. Randy, who does autopsy all the time. Probably, let's see if he happened to see some of the graft. Thank you.
1 o'clock, so thank you everybody for your attention.
Thank you. about the timing thing. It's
I
always
know, I understand that you're traveling around that time.
right.
I travel.
So do they allow us to do?
I think they would prefer people to be in person.
Yeah.
But I think Kevin is hoping that we can make it happen during the first week of May.
Oh, okay.
I think the program would rather postpone to first week of May.
Okay, and make me the back person because I was traveling. She cannot do it.
But yeah, no. I mean, everybody's always on that.
Okay.
So
Okay.
there's been times in the past years where
Okay.
other people have to do it.
Okay.
But yeah, hopefully we can find a day.
Yeah, we
in
should.
the first week of May.
Yeah, and
that
also.
Joe also can. So
Oh,
have
good.
you met Joe Alston yet?
Yeah.
Yes.
Yeah.
Yeah, he's great. So I
Yeah,
was like,
yeah,
I
he's
was
good.
really excited that you and Joe get to go see us.
Yeah, we
And Alex also, of course. But you guys are the best. I mean, they're
we're the
really
few basket people. Yeah, right.
So.
Okay, good seeing you. Yeah.
Thank you.
Okay.
Really fun talk.
Okay, thank you. Hi.
I know you're looking. I'm Ellie. Nice to meet
Oh,
you. I'm in
good to
Matt's
see you.
lab. My husband is a postdoc in Nabil Al-Qayyum.
Oh,
I don't know
good.
if you've reached out to them about collaborations yet, but he looks at parasites in diabetic stroke.
Who is that?
Dr. Nabil Al-Qayyum.
I know. Who's your
Oh,
husband?
Bailey Whitehead.
Oh,
He's a
okay.
postdoc there.
Because I know Paul.
Oh, yeah. He works with Paul.
Yeah, Paul is the one I supported for his. I guess I'm a mentor on his AHA grant.
I knew you were looking for collaborators, and I
Yeah.
was like, let me just mention it.
Good. Yeah, that's great.
Great.
Good.
Listen,
Okay,
great
I will.
talk.
Oh, thank you.
Yes. Okay, yeah. So, yeah, it was nice to see the actual
Yeah, we talk all of right.
Yeah, we met
Mm-hmm. Yeah, thank you.
she's here, we haven't made time to hang out, so we should totally make time to hang out with Randy.
Definitely. Yeah.
Well, I think we'll come up with lots of ideas.
We'll
We'll see, like NIH catches up.
Oh, that's right. Oh my God.
Well, at least I'll have
Well, I mean, like, it's kind of funny. I'm like, oh, isn't it fun that we're going to be excited times again? I was like, plans are paused, everything is paused.
Oh, did any news today say they're going to reverse?
Yeah,
Oh, no, yeah.
Rescind it. Okay.
Yesterday afternoon, the judge had paused the pause,
Right.
and by this morning, they had rescinded the pause. I mean, like, we're back to class times.
Great ideas.
Oh, thank you. I'm Steve.
Okay.
Nice to
Okay.
meet you.
Who has who
The
has?
sponge model?
Yeah.
Okay, all right. Well, I know about this because I consulted for a company that did lots of that. So I learned about it, not from my own lab work, but I was serving as a consultant for a company that was targeting tumor vascularization. And it was an interesting story because they were actually trying to figure out what was it when women are giving birth that have a group B strep infection, that the toxin is then passed to the baby, and so that's the reason every single birth, the moms are being checked for group B strep. And it's because the vascularization in newborns has receptors for the toxin that the group B streptococcus is producing. As we age, that we lose the expression of the receptors that bind the toxin. And what they discovered was that the neovascularization to a tumor re-expresses the receptors for the group B streptococcus.
Is that right?
And therefore, you can give someone this toxin. Our normal vasculature
We're not,
and
but
only the neovascularization
oh, that's cool.
to the tumor. And so what they did was is all neovascular, because if it's all neovascularization
Then-
versus
then-
tumor-specific,
yeah.
you don't want to do that. And so what they did was that in animals where they both had tumors and they implanted the sponge in the ear, that the normal vascularization that was being propagated either from a wound healing
Or by the-
or from the sponge went on unaffected, while they were still
Only the tumor.
selectively killing the tumor.
Oh, that's
the
nice.
vasculature going to the tumor. So they developed a program where they said, we're not going to try and kill tumors, we're going to kill their blood supply. And so
Yeah.
NIH, in all of their wisdom and study section wisdom, rejected this guy's work for 15 years. He took it on the road, and China is now developing this, and it is incredibly
That's another
effective.
evidence China is bad.
It's amazing. He ended up moving his whole operation because he demonstrated proof of principle, and they said it all just makes so much sense.
Oh
and
my
so he's
God.
moved his entire operation. This is when I was at Vanderbilt, and he was in the Department of Biochemistry at Vanderbilt studying. He started out.
When did you move here?
20 years ago.
Oh,
so
okay.
my knowledge is very old. But,
Similar
but it
to
was
Randy.
But the wound healing stuff. So I have one more thing. Very few seminars do I write this many questions.
Oh, good. That means I didn't make it clear.
I took no. My questions are, if that, then this ought to be true. It's not. I need clarification. So it's piecing together parts. But
That's okay. We're on the line.
We're going to lunch.
real quick. So a friend of mine at the University of Alabama in Birmingham,
Who is that?
Scott Ballinger.
Oh, he's my buddy.
You know Scott?
We're in the same department. We were.
Oh, you missed the very beginning of the introduction.
Yeah.
Oh my
She was
God.
the vice chair of pathology.
I was a vice chair for faculty development there.
Okay,
Yeah.
all right. So Scott was a so after Vanderbilt, I moved to UTMB.
Okay.
Scott was across the hall in Ben Van Houten's lab.
That's right.
Okay.
Yeah,
And
yeah.
so I was in the same institute where Ben Van Houten and
Okay.
all kinds
Yeah,
of other
yeah, yeah.
people. Okay, so you know Scott.
Scott's work, the mixed mice. Yes. We did that for our study as well.
Okay, and because
And Scott is not doing research.
I'm a believer that if we improve our mitochondrial DNA
Yeah.
repair and mitochondrial
Me
health,
too.
we'll all live to be 1,000.
Not necessary, but any part of our work. Yeah.
But in terms of so he did the mitochondrial
Switch. Yeah.
exchange in
CH3.
ApoE deficient.
Yeah.
Okay. And the last time I followed the story, and it's been quite a while.
And I should be on the paper of Alex something which was the last time modified 5 years ago, but still not published, I think.
Well, but so in terms of then, if you do the mitochondrial swap, then, and I'm wondering is that if you could do that in your RUNX2 knockout, I was just wondering how that might modulate.
It could, you mean mix the bad mitochondrial DNA into my RUNX2 knockout? Yeah. And that's a good question. We haven't, the crosstalk between.
That's probably a real tough reading.
Yeah.
I looked at that reading scheme, it looked like a nightmare. So, okay, all right, so. Sorry.
So we can keep talking and just shoot me an email. Or
Okay.
do you have my email?
I'm sure you're.
I
easy
will.
on it.
Make sure.
Okay, okay.
So,
So I think we need to think about the question you had. And And definitely the wound healing part, I want to continue because Joanne Murphy Oric, you probably didn't know. She's the one who did all my wound healing
Oh,
part.
okay, okay.
But she's retired now.
I see. Well, I'm sure it's, I believe, a really simple
It worked.
surgery.
It were.
Super simple.
It's very straightforward. It will at least give us some kind of thoughts on why the male-female thing, and also fibrosis versus calcification. So that's good.
Okay.
Thank you
And
so
at some
much.
point, so I'll also just say, I'll send you a little bit of information. If you had a mouse that you put on a high-fat diet and it gained absolutely no weight, did not get fatty liver disease,
That's
did
great.
not get dyslipidemia,
Yeah,
would you be interested in
I
that?
will be very much interested.
Okay. So we have
And,
mice that overexpress a mitochondrial DNA repair protein. You can put them on a 60% fat diet and they gain no weight. Their livers do not become fatty, etc.
Okay, let's talk about that.
Okay.
I will be very interested. Any animal models with phenotype, I will be. Yeah.
This is a series. I'll send you a couple papers.
Okay. Okay.
Okay.
All right.
Great. Thank you.
Okay. Great. Thank you.
Hey, Matt.
Yes.
Keep inviting speakers.
That's not good. Did that disappoint you?
I guess I'm supposed to invent more good speakers like you. Is this yours?
I think it is. It's not mine. I think it belongs
Okay.
here.
So I'm just gonna leave this here.
I
But you
guess.
should take your
Oh, the thing.
that off,
And they needed to quit WebEx.
and then I'll just plug that back in. Not really sure what I'm supposed to do here, but I'll just